Hello, friends. How you doing today? Okay. Can I hear a little bit more about how you're doing? How you doing today? Okay. Welcome to the John G. Batsakis Lecture at the 11th Annual Clinical Pathology Symposium. I'm Karen Barron, your Allied Health Education Manager. On behalf of Dr. Valdez, Christina Martin, the Planning Committee, and the exhibitors we have here today, give it up for our exhibitors. We're very glad that you're here to learn and connect and celebrate the amazing work that we do every day. The sessions at today's CPN Symposium are approved continuing education in the clinical laboratory sciences by the ASCLS PACE program. PACE-approved programs are accepted by ASCP Board of Certification for CMP credit, recertification credit. The CE organizer may be used to document attendance, obtain a certificate, and claim credit for participating in these sessions. And you can get a flyer at the check-in desk about how to use the CE organizer. Both ASCLS members and non-members may create an account with CE organizer. At the end of this session, I'll give a unique session code that you can use to claim credit. And instructions are available. The deadline to claim credit through CE Organizer is May 9th. After May 9th, though, you can still put this activity in directly into your CMP. Today's sessions, you'll also get credit in the UMich Cornerstone Learning System, so make sure that you've stopped at the check-in desk to make sure that happens. I'd like to introduce Dr. Ricardo Valdez, Director, Division of Clinical Pathology and Clinical Laboratories, and the John G. Batsakis Professor of Pathology.
Thank you, Karen, and welcome to everyone again. Thank you, Karen, for the introduction. I was going to do that myself in case people didn't know me. Since 2017, our keynote lecture for this symposium has been named in honor of John G. Batsakis. Dr. Batsakis pioneered and grew the discipline of clinical pathology in our department from 1950 to 1980. In 1980, he left here to become the chair of pathology at MD Anderson. He is credited for establishing the clinical chemistry laboratory, as well as for recruiting pathologists and lab staff who ultimately went on to establish many of our specialized laboratories, including microbiology. This year, it's my pleasure to introduce Dr. Aubree Gordon as the John G. Batsakis CP Symposium lecturer. Before calling Aubree up to the podium, I'd like to tell you a little bit about her. Dr. Gordon received her Bachelor of Science degree from the University of North Carolina at Chapel Hill in 1998. She subsequently headed out west to California, where she spent several years at University of California, Berkeley, receiving an MA, an MPH, and a PhD in 2009. She was recruited here in 2014 to the School of Public Health, where she is currently an Associate Professor of Epidemiology and Global Public Health. She is also the director of the Michigan Center for Infectious Disease Threats and Pandemic Preparedness, something very applicable to today's talk. While her expertise is broad, her research focuses on arboviral diseases and respiratory virus infections, such as coronavirus or SARS-CoV-2, but also influenza. Dr. Gordon's work examines both the transmission of respiratory viruses and the development of immunity. She is a recognized expert and has published well over 100 papers in her field of expertise. As part of her work, Dr. Gordon has established a number of large field studies of influenza and/or SARS-CoV-2, including a prospective pediatric cohort study in Nicaragua, a family cohort study in Nicaragua, and an adult cohort study in Michigan. In addition, she also led the establishment of the Immunity Associated with SARS-CoV-2, or IASO, cohort study, and she's here today to present findings and observations from that study. Please join me in welcoming Dr. Aubree Gordon to the podium.
Can you hear me okay? Yep. So I wanted to thank Rick for that very kind introduction. And I also just wanted to thank the organizers and everybody for giving me a chance to come here and speak today. I'm going to be talking about IASO, and IASO is really a pathology study, so I feel like I'm a little remiss actually that I haven't come to present it before in pathology, so thank you very much for this opportunity. So IASO, Immunity Associated with SARS-CoV-2, cohort study, as the name suggests, was set up really to address SARS-CoV-2. IASO happens to be the goddess, the Greek goddess of recuperation from illness, and that's where the name came from. We were challenged by the NIH actually to come up with a Greek or Roman name, and that one seemed particularly fitting. And it was actually originally suggested by my, at the time, 7-year-old daughter, who was like, well, could you think of something for IASO? Because I think that would be great. So there you go. So I'm going to take you back with me to 2020 and the first half of the year. You know, when SARS-CoV-2 was first spreading in the United States and first really spreading throughout the world. And just to kind of highlight a lot of the unknowns that we were facing at that time, really from like a scientific perspective. So we didn't know a lot about the natural history of SARS-CoV-2. Certainly that information was coming pretty quickly, but there were a lot and still remain a lot of questions to answer. We didn't understand how transmission occurred at that time, right? I'm sure a lot of you can remember like people wiping down their groceries, right? Because we really weren't sure how that was occurring. We didn't know about immunity, how people would develop broad immunity, hopefully, to SARS-CoV-2, whether or not reinfections would occur, right? If you think back to 2020, that was a big question. How protective would an infection be? And then about viral change. We weren't sure how much coronaviruses changed. We weren't sure if we would have variants or not. Of course, we all in this room know the answer to that question now, right? The answer is yes, but in fact, the scientific community got pretty upbeat about it right around August, September, I think, is when we started going, you know, I don't think this virus is going to change, and then of course, rapidly it did. So when we think about all those unknowns, and you think about like, how do you go about studying a new disease? There are a lot of different study designs that can be used. These are the main study designs. Does that work? Well, can you see the pointer? Here, how about this? Laser pointer. All right. So one of the main study designs is a cohort study. And cohort studies can be used for infectious diseases. They're used for other diseases too. Most of these designs, in fact, other than the case ascertained study, can be used for non-communicable diseases or often used for non-communicable diseases. So in a cohort study, the basic study design is you take a group of people and you're going to follow them forward through time. And so you assess their exposure or exposures, depending on what you're looking at. And then you're going to follow them forward for your outcome or outcomes of interest. Right, and so, you know, for our study, our main exposure would be different immunological factors, right, neutralizing antibodies, T cells, things like that, and we're going to follow people forward to see how they're protected or potentially not against SARS-CoV-2 infection and illness if they do get infected. Another study design that you can use and is often used for infectious diseases is the case ascertained study. Sometimes called an index cluster study. And in this study design, what people do, it's a shorter duration, right? So they identify people who have the disease. So these were often done for flu. I do studies like this for flu myself. But basically, you wait for people to come into a clinic and you're like, oh, you have influenza, right? And of course, they're gonna go home. So they've got a group of people around them that are probably gonna be exposed to flu, right? Because they're not going anywhere. So you go and you enroll all those people, often in a household, but not always. And you're going to look at factors that affect transmission, things that affect susceptibility of individuals to either getting infected or, if they do get infected, to them getting sick. Another study design is a case control study. So case control studies kind of start here, right? So you identify your cases, and then you identify controls. And then you're going to look back and kind of say, like, well, before they had whatever my disease of interest is, what was, various exposures. Case control studies are used a lot, particularly at the beginning of an outbreak for infectious diseases. So do you all remember Zika? Right, probably. Yeah, I worked on Zika a lot. I worked on chikungunya because I work on arboviral diseases. And so case control studies were really pivotal in figuring out what was going on with Zika, because people down in South America said, wow, we're having a lot of babies with microcephaly being born, and they had no idea what was causing it, right? So then they defined their case, enrolled controls, and started to look at a wide variety of exposures. And so case control studies are particularly good for questions that you need to answer really quickly, and particularly where you know what the disease is, but you have no idea what's causing it, then a case control study is a really strong study design for that. They're often also used in foodborne outbreak situations. And another study design is a cross-sectional study. So in a cross-sectional study, kind of think of your classic survey. You're taking a snapshot in time, right? So I could go around and ask everybody in this room questions on a survey. That would be a cross-sectional study. You're all lab people, right? We're all lab people. I am too. And so another cross-sectional design that you see a lot of, or we've seen a lot of during SARS-CoV-2, are serosurveys, where they go through and they'll take kind of a swath of the population and measure their antibodies to try to figure out what's been going on with SARS-CoV-2 and to model risks going forward as well. So these are kind of the standard study designs that you think about when you start to study a new disease. And we had been asked specifically to look at immunity to SARS-CoV-2. This is 2020, so pre-vaccine. So specifically, I was asked to look at correlates of protection that came from infection, right, because we didn't have any vaccines yet, with an eye towards the vaccines are coming. So please set up a study that could also look at vaccines as well. And one of the things that I did, or still do, but did a lot pre-pandemic, was look at novel correlates of protection for influenza, kind of with an eye towards, well, not even just kind of, it actually is what I do, right, with like, what would we ideally like a vaccine to do, right? We've probably all had the flu vaccine, yes, or a lot of you have. You've all, either yourself or known other people who got the flu vaccine and still got the flu, right? We know that the flu vaccine, it's a great vaccine, but it is suboptimal. We'd like something that protects better against infection. And I think at this point with SARS-CoV-2, we can say the same thing, right? And so there was a kind of a little bit of, an eye towards, we don't know how good these vaccines are gonna be. They're probably gonna be a first gen, and they turned out great, in fact, right? This is probably gonna be a first generation vaccine. So we want you to set up a study where you can really think about novel correlates moving forward. So things in addition to the immunity that's being generated through the vaccine. So that was kind of the request that came to us, and we went with a cohort study, in part because it's my favorite, my favorite study design, but also in part because it is really the main study design, I would say, to look at these sorts of questions long term. So cohort comes from the Latin word cohors, which is a group of Roman soldiers, so it was one unit of the army. And I always find this helpful because then when you think about cohort studies, you think about people marching forward together, right? So in this case, for cohort studies, people are going to be marching forward in time together. So some of the benefits of cohort studies and why we selected this design for IASO is temporality. Because you're assessing exposure first, and then whether or not somebody becomes whatever your case definition is, you know that exposure happened before they became a case, right? So that's a big strength. Another huge strength of cohort studies is that you can actually study multiple exposures and multiple outcomes in the same study. Whereas like with a case control study, you have to decide what your definition of a case is, right? So that's your one outcome. You can't decide you're going to do a different outcome with a case control study. But with a cohort study, you can do all sorts of diseases and outcomes, potentially, of course, where you are focused in on SARS-CoV-2. But when I think about cohort studies, an example of that, you might have heard of the Framingham study or the Nurses' Health study, maybe some of you have, right? Those are studies where they've looked at hundreds of different diseases, hundreds of different exposures, really pivotal studies. Cohort studies are also sometimes the only study design to do when you're looking at exposures that are not typically assessed or known. You know, so if I went up to any of you and asked you what's your SARS-CoV-2 antibody level today, and what was it 3 months ago, you probably wouldn't be able to give me that information because you're probably not measuring it all the time. And certainly if I started to ask you about your T cells, you're like, well, I mean, I don't know, right? And so we don't know what's going to happen to people in the future, right? So you measure those outcomes, those exposures, and then follow them forward. And we're really looking for things that are going to protect people both from infection and from disease if they're infected. So that's why we decided to go with a cohort study. So there were some real challenges and opportunities that we were facing back in 2020. One of the big challenges was that the university wanted us to have little to no contact with participants. So I don't know how many of you have worked in a study, or probably some of you are in IASO, so you've been in a study, you may be in other studies, right? But normally how this would work, or at least in my prior experience, is that we would have you come in if you were going to be a participant. You do the screening, or you might do the screening ahead of time. We'd set up an appointment. You do the enrollment surveys. We'd give you information about we'd do the consent. We'd give you information about the study. You do the enrollment surveys if you signed up. We'd collect your blood samples and various samples right then, and we'd kind of do it all in one, right? This wasn't an option in 2020. We had to kind of rethink how we were going to enroll people, how we were going to get all of these things done. Sorry, the slides are slightly off a little bit, but it's okay. There was also a lot of hesitation, for good reason, on potential participants' parts, right? People were like, well, I don't know. I mean, I am social distancing. I don't have contact with anybody outside of my house, and you're going to ask me to come in. You know, I want to be helpful, but I'm going to have these extra exposures. So we had to really think about a way where we could make sure that we were helping to answer questions about SARS-CoV-2, but certainly not helping to spread SARS-CoV-2, right? And really making participants feel comfortable. So that's some of the challenges we were facing, but there were some real opportunities as well. Motivation. The U of M community was amazing in how much people wanted to help. People were like, sign me up. You know, I'd love to help answer this question. And so we had an incredibly motivated population. And you'll start to see that when I go over kind of recruitment and how many people we actually talked to that qualified for the study and how many signed up. U of M Health System and the labs here were a huge strength and really came in to kind of save the day for us to be able to set up this amazing study, right, which is a partnership between pathology and the School of Public Health. So IASO, immunity associated with SARS-CoV-2, was established to address SARS-CoV-2 immunity. As I already said, it's a partnership between the School of Public Health and Michigan Medicine, specifically pathology, and it's NIH funded. It's a prospective U of M employee-based cohort study. So to originally be enrolled into the study, you had to be a U of M employee and working in person at least 2 days a week, so be an essential employee. That was, once vaccines started to roll out and become really readily available, that was widened to any U of M employee. And then later, when people started to retire, who were in the cohort, individuals that retired and were in the cohort were able to stay in the study. But predominantly current U of M employees. We monitor for SARS-CoV-2 infection We look for both symptomatic infections and asymptomatic infections. So for symptomatic infections, we use participant self-report and do testing on respiratory specimens. And then for asymptomatic infection, we are using like results from blood tests. And then we monitor the immune response to vaccination and infection in these individuals, and really that's done with blood samples that we collect through the study. So this is the basic study design. So it's a cohort study, right? So we've got this group of people that we enrolled. We follow them forward in time. We have a number of surveys, which I'll go over, that we collect from all participants. We get a blood sample every 2 months. We also collect blood around vaccination. We collect blood around infection. And when people do get infected, at least on a subset of people, we try to get them to come in for serial sampling. And this will allow us to address questions about things that might affect transmission, right? Because the shorter the duration that you shed virus, the less likely you are to spread the illness, right? In addition, the level of shedding we know is also important. And so for flu, we've been able to look at specific immune markers and correlate those with protection from duration of shedding and level of shedding. So kind of hoping to do the same thing with SARS-CoV-2. So for the study components, we've got the surveys, right? We collect blood samples, and then we collect respiratory samples from all the participants when they have symptoms, or if they don't have symptoms, but they tested and figured out they were positive, then we'll collect respiratory samples too. We try to get respiratory samples on everybody. That's becoming a little bit more challenging with home tests, because people just want to test at home, but we really like to do it because we like to confirm the infection. We also like to know, ideally, what virus people were infected with, because that's really important moving forward. So for surveys, we collected a baseline survey at enrollment, lots of demographic information, health information, et cetera. We then got that survey at each re-enrollment. People generally, they enroll in the study for a year. We have a bi-monthly social distancing survey that asks questions about how often people work in person, if they social distance. At one point, it's greater than 90% of the cohort reported that they did some level of social distancing, which was different for each person, right? Now it's a much lower, I'd say less than 10% of the cohort, I think, reports social distancing, but we still ask those questions. Really important for characterizing risk, right? Because for some things, like for people who uptake the bivalent booster, for example, they actually are a bit more cautious in general. It has kind of two effects. One is that they might be less likely to have exposures because they're still going out less, they're wearing masks more. On the other hand, they're much less likely to have ever been infected, and that's also protective. So you have to kind of think of these things when you start to think about measuring vaccine effectiveness. So we have these social distancing surveys. And then we have weekly symptom surveys. They're quite short. Usually they're 2 questions, except if you report symptoms or testing, and then there are additional questions that come. When people get infected, we have what we call a seroconversion survey, also used for when they just report an infection. So it can either be a blood test or a reported infection, but ask about that infection. Did they know they were infected? How did they know? Do they have an idea of how they might have been exposed, and ask about symptoms. And then we have a post-acute sequelae of SARS-CoV-2 infection, so long COVID asking about any long-term symptoms that people get at 30 days after infection, and then every 30 days thereafter until they report that all of their symptoms have resolved. So for other data sources, in addition to those that come kind of directly from the participant, we also can access medical records in case some infections aren't reported to us. We get information from the vaccine registry, which we check with the participant reports. And then the participants themselves will contact us with information, so they'll email us or call in to let us know what's going on. So for recruitment began in October of 2020. We originally, when we were planning the study, we thought we weren't going to call anybody. The IRB let us know that, in fact, we would be calling people. And so we ended up making almost 6,000 phone calls to reach about 4,800 people and did this pretty quickly. Of those 4,800 people, 76% of the individuals were eligible to participate in the study, and so they were sent a consent form. So 3,721 people. About 10% did not consent to participate in the study. That's actually a really high uptake. Normally you would send consent forms and maybe 10% might agree to participate in the study. And so that's where that motivation really came in. In total, 3,356 people enrolled into IASO in the first year. We also did a year 2 and year 3 enrollment. So looking at the total number of participants over time, you can see we kind of had a little bit of a slow start. That was on purpose to make sure all the systems were kind of up and running. And then we rapidly ramped up and enrolled about a little over 3,000 people in about 4.5 months' time, so quite quickly. Our participants range in age from 18 years to 94 years of age. But you can see here, and on the next slide actually, that a majority of our participants are between 30 and 60 years of age. We also are heavily slanted towards being female. There's probably a number of different reasons for that. 86% of the cohort identifies as being white, 7.3% Asian, and 2.7% Black or African American. About 2% are identify as more than one race. So for those surveys, we've sent out over 300,000 surveys now, with an overall completion rate of about 96%. This is slightly off. So really a ton of surveys. Those weekly symptom surveys make up a huge bulk of that, right? Because each participant is getting those every week. So that's kind of the setup of the study. I'm going to briefly talk a little bit about SARS-CoV-2. People often lead in with this, but it's not necessary at the beginning of the talk. So humoral immunity against SARS-CoV-2. When we talk about humoral immunity, we are talking about antibodies to SARS-CoV-2. As you probably already know, the SARS-CoV-2 vaccine targets the receptor binding domain, the RBD, of the spike protein of SARS-CoV-2. This is SARS-CoV-2 over here. So we've got that spike protein, which is what gives the SARS-CoV-2 virus its name, the coronavirus, right? But infection, on the other hand, so vaccination, really just targeting the spike, RBD. Infection, on the other hand, you're exposed to the entire virus if you get infected. And so you'll generate antibodies not only to the spike, but you'll generate antibodies to various areas of the virus, including the nucleocapsid protein, which is here inside the virus, right? So you get those antibodies when you get infected. If you've been vaccinated, you haven't been infected, you won't generate those antibodies. So we, through this study and really through pathology, have collected over 26,000 blood samples from IASO participants since that began. I know Rick and Carmen are always they're like, yeah, it's not that many samples, right, when we talk about them on a monthly basis. But for a research study, this is just a huge amount of samples to be collected. So they're collected and they get tested in the special chemistry lab for spike antibodies. So here we can see over time, in the beginning, a lot of individuals had no spike antibodies, but we had that rapid ramp up of vaccination, where almost all of the cohort then had spike antibodies, predominantly from vaccination. On average, participants have had 4.75 blood draws per year, and overall, we have about a 76% completion rate for those bi-monthly draws. So we're looking, to know when people get infected, right? And we were also looking to characterize the difference between immunity generated by the vaccine and through infection. And so here in the blue is spike antibody levels, so spike titers about 30 to 60 days post infection, and then over here in maize, spike antibody titer is about 30 to 60 days post-vaccination. And one of the things you'll notice right away is that generally speaking, if you get vaccinated compared to if you were to get infected, right, and not be vaccinated, the vaccine tends to produce more robust antibody titers than vaccination. However, there is a lot of overlap between those distributions, so you can't use spike antibodies alone to differentiate infection, and that's particularly true in vaccinated people, particularly if they've been boosted, because what we'll often see is when somebody gets infected, we don't see much of a rise in their spike antibody titers, and that's because they've kind of hit a threshold level, and at least there may be changes, but our test is not detecting those changes anymore. The other thing which I think is pretty important to note here is in spite of these massive differences, these are first infections, by the way, when you get if you get a second infection, or when people get second infections without vaccination, their titers do go higher. In spite of the big difference in these antibody titers, so they both correlate with protection. So antibodies from infection correlate with protection. Antibodies from vaccination correlate with protection, but the level of antibodies that you need to have like 95% protection in the first year of the pandemic following following vaccination was significantly higher for vaccination than infection. So people would have much, much lower levels of antibodies, particularly neutralizing antibodies, and yet they had about 95%, 93 to 95% protection against reinfection, so very similar to the vaccine. One thing that tells us is that in immunity that is infection-induced, there are other protective factors there, right? And that's one of the things we're after in this study, and so I just kind of wanted to highlight that. All right, so in order to detect infections, in part, we use that interior protein, nucleocapsid protein, or specifically antibodies to that interior protein. So following Omicron, we did a big push in the study to get an NP antibody test on everybody that was in IASO, really to see, because there was this theory going on, like a lot of people were saying, like, you know, like, the infection rates are really high, but the true infection rates, you know, are 10 or 20 times what people are reporting because there's so many mild infections going on. So we were like, well, I mean, we can look at that, right, and see. The answer, at least in our study, was like, no, it wasn't, right? Certainly there were asymptomatic or subclinical infections going on, people who weren't reporting them, but most people who didn't think they had been infected, in fact, had not been infected during that first Omicron wave. We've also been using in antibodies to confirm infections. We want to know that people were really infected, and you know, there can be some error in lab tests. There are errors certainly in antigen home tests. I have a friend who one particular brand of test, who knows why, every time she takes it, it's positive. And although I've told her, like, don't buy that brand. Like, that one doesn't work for you, right? Like, I don't know what it is, but to like, stop it. You don't have COVID today. You know, it's just whatever. So anyways, so we as much as we can, we try to get 2 lines of evidence for each person. So that could be 2 respiratory tests, potentially the same type of respiratory test on different days, respiratory test and antibody, right, to really know that indeed confirm that infection occurred. Here we did another kind of in-screen on individuals in the study who hadn't reported infection and who we hadn't previously detected an infection in them serologically. It had been kind of a little while since we looked, and what we found was about 14% of those individuals actually had been infected between here, which is kind of June of 2022, and this about a year later. So for SARS-CoV-2 infections over time, we've had 1,713 infections in the cohort study. The beginning of the study, almost all of those infections were first infections. You can see here there are few, but in the fall of 2021, we start to see a little bit of a pickup of second infections. That's really when Delta started circulating, here a little bit with earlier variants. But we really start to see more second infections start to occur in the cohort with Omicron coming in, we're now starting to see some third and fourth infections. Looking at this another way, we can look at the immune status of people in the cohort over time. So in the very beginning, right, almost nobody is vaccinated, or they're partially vaccinated here in January 2021. We start to see that vaccine rollout here in light blue. These are people who are vaccinated and have not been infected with SARS-CoV-2. This kind of gray-blue color here are individuals who were infected prior to vaccination. And then in the darker blue down here are individuals that were infected after vaccination, and you can kind of see that shift happening over time. We also have individuals who did not get vaccinated, and you can start to see infections accumulate in them. And I think back in October of 2022, by October 2022, everybody in the cohort had immunity to SARS-CoV-2, which was either through vaccination or infection. So looking at infections by immune status, right, this has historical infections too, so infections that happened to people in the study before they were actually enrolled in the study. So obviously in 2020, all of those infections were occurring in unvaccinated people because the vaccines hadn't rolled out. We started to see infections in partially vaccinated people in January, and then kind of this pickup in fully vaccinated. And this is, remember that the cohort's changing too, right? Like people here, U of M mandated the vaccine. In general, the community was very receptive of the vaccine regardless of the mandate, so people kind of ran out to get vaccinated, or majority of people did. And the same thing with boosters. Booster uptake was fairly heavy in the cohort, and so we started to get a lot of cases of breakthroughs in vaccinated boosted people because a majority of the cohort at that point was vaccinated boosted individuals, and then more recently with vaccinated boosted with the bivalent booster. So I'm going to slightly shift gears a little bit and talk about IASO's role nationally and really internationally in helping to monitor SARS-CoV-2. This is a slide on SAVE. So SAVE is the SARS-CoV-2 Assessment of Viral Evolution Group. It's a risk assessment pipeline that was set up to assess the impact of SARS-CoV-2 variants. It was set up to give information to the U.S. government, to this group called the SARS-CoV-2 Interagency Group, or SIG. I will not be able to list all the agencies that are part of that, but CDC, BARDA, FDA, NIH, HHS, the Department of State, the DoD, et cetera. Basically, kind of everybody coming together to make decisions about how to distribute vaccines, updates for vaccines. We played a role in determining what strain should go into the bivalent booster, and giving public health information to state and local governments as well. So SAVE is a coordinated team of academic and U.S. government scientists. It's got expertise in computational and structural biology, molecular biology, immunology, virology, genetics. I somehow didn't put epidemiology on there, which is what I am, but epidemiology as well. Tasked with identifying variants with concerning properties, then to define assays and experiments that should be done on those variants to characterize the risk, generate that risk assessment data, and then share that data in real time with other members of SAVE and with the U.S. government. And then that data is actually also shared with other governments as well, such as the UK. Neutralizing it. So when we talk about antibodies, you probably heard a lot about neutralizing antibodies, I would guess, over the last few years now. But neutralizing antibodies are just one type of antibody that your body generates. We generate a lot of binding antibodies. We generate antibodies that might interact and activate complement systems. But here we're going to be talking specifically about neutralizing antibodies. And neutralizing antibodies are antibodies that bind to a virus will prevent receptor binding, and so then the virus cannot enter the cell. And the reason we care about neutralizing antibodies so much with SARS-CoV-2 is neutralizing antibodies have really been shown to correlate with protection. It's a great paper in Nature Medicine showing the various vaccines. I think this is Moderna up here and Pfizer right there, that's maybe Novavax, and how the level of neutralizing antibodies that those vaccines generate really correlate with vaccine effectiveness. Neutralizing antibodies for infections generated through infection have also been shown to correlate with protection, although lower levels are needed. And so neutralizing antibodies really provide us a way to look at variants and see how they might escape pre-existing immunity, both immunity that's induced from vaccination and immunity that's induced from infection and hybrid immunity. And so one way that we look at neutralizing antibodies is through neutralization testing. So it's a laboratory technique used to determine the ability of antibodies to neutralize a specific pathogen. In this case, it'd be SARS-CoV-2. So FRNT is one way to do that, focus reduction neutralization test. So we've got a sample of SARS-CoV-2 virus. So it's live virus, right? It's going to be exposed to a dilution series of serum. And that serum has antibodies against SARS-CoV-2. So you've got high concentrations here, the concentrations go lower, and then you're going to count those dots, right, which are the focus-forming units. So when the, so you'll count the dots, which are the focus-forming units. So when you see a reduction in the focus-forming units, it allows us to look at how much change is happening. So you've got high concentrations of serum here, right? So it'll neutralize the virus, so we get very few focus-forming units. But as we reduce the serum concentration, you get less neutralization of that virus from that serum, right? And so then those cells don't get infected and they're maintained. So what you can do, or what we do to look at SARS-CoV-2 variants is we'll take, this is like early ISIT, could be a virus from 2020, right? Maybe we're using serum from somebody who's had 2 doses of the vaccine, of the mRNA Pfizer vaccine, or 3 doses of the vaccine, right? And we're gonna look at what does the original virus look like? What about variant 1? Maybe it could be Delta, right? What about variant 2? Maybe that's kind of BA1, the original Omicron, right? And we can see how much that virus has changed, how much it's allowing it to evade neutralization, which gives us an idea of how much protection people are gonna have against that variant. And we can, of course, use serum from different people who are infected, people who are vaccinated, different vaccines, different series of vaccines, to get a better sense about how both the vaccine is functioning and how those variants are changing. So there's another way you can do this. So that way is fantastic. The only problem is, has anybody in here ever worked in a BSL-3 laboratory? Yeah. So all of that work has to be done in a BSL-3 laboratory, right? There was another method that had been set up, I think, used for HIV, some other viruses before, called pseudovirus neutralization. So that's another way to really look at neutralization, except in this particular case, you are not using live virus. So it doesn't actually, so it's not infectious, which means you can do it in a BSL-2 lab, which is really nice. So you do that by taking the envelope or the outer proteins of one virus and putting them basically onto another virus, and makes it non-infectious, means we can now do it in a BSL-2. There are some caveats to that, right, because you're not really looking at the entire virus anymore, but fairly early on in the pandemic, it was shown to give kind of similar results. And so IASO collaborates with people who we do both of those with samples to look at neutralization. So using pseudovirus neutralization, you can address emerging variants. You can also do things looking at like study viral entry into cells. So in pseudovirus neutralization, we're not going to be counting the cells anymore. You have the pseudovirus here, then you infect the cell, right? And if the cell gets infected, it's going to turn on this reporter gene called luciferase. It's luminescent, right? Basically, you read how much luminescence you get, lights on, that virus, that pseudovirus, was able to enter the cell. On the other hand, you can add antibody or serum. If it neutralizes the virus, you won't get infection anymore. You won't get it entering the cell. You won't get that luciferase getting turned on, and so you get that lights off, right? So that's another way to do neutralization without using live virus. IASO has been participating since pretty early in 2021 to really look at how variants are changing and how they may or may not be evading immunity, specifically neutralizing antibodies. So over here, we have, these are actually both papers that were done in collaboration with Yoshi Kuoka and his group in Wisconsin, at the University of Wisconsin, Madison. Here, looking at the Delta variant, over here, Gamma variant, but once again, looking at how these variants are or are not able to evade immunity, so we're looking at a fold reduction in neutralization. We've been also doing that more recently with newer variants. Here, a pretty recent paper. This is using pseudovirus neutralization, looking at BQ1 and XBB. So basically, as soon as the way SAVE works is these variants, there are a bunch of people looking at the phylogenetics, kind of identifying, what variants they think might be risky. Try to get an isolate of that virus. If not, they recreate it. Essentially, pseudovirus, they can just do that from sequence, right? Gets distributed through the network to various labs. We then work with those labs to figure out what their needs are in regards to different kind of immune profiles, right? And then we will look at how that variant might evade immunity or not. And then that information goes in with other information because people are looking at how it transmits, right? And how it transmits in animals. So there's in vivo, in vitro experiments going on to put together a whole risk profile and pass that information on to the US government. We also worked on monitoring vaccine response. So here is data looking at, you can see up here, 3 shots of the wild type, either our mRNA vaccine, 4 shots of wild type, or 3 shots of wild type plus a BA4 or 5 bivalent booster. And then here it's not showing up that well, but it's a BA4 or 5 breakthrough case, so somebody's been vaccinated and then gotten infected afterwards. And what we're looking for here is to look at the fold reduction and neutralization. What we were really looking for or hoping to see, I can't see it very well here, sorry, is for BA4-5 right there. We wanted to see not that much of a fold reduction compared to the original strain rate, which is right here, D614G. But you see several fold reduction, right? It's lower. It's very similar to 4 shots of the original vaccine, meaning that we didn't see that the bivalent booster generated more immunity to BA4-5 than the original vaccine. However, I would say it is significantly higher than what you see with 3 shots of the wild type. So while we didn't see a clinical difference, or what we would expect to be a clinically significant difference between the monovalent and bivalent booster, if you're using them as a 4th shot, both of those did generate robust immunity compared to not getting a booster. We then asked the question here, we were kind of wondering, so if you're exposed to something and you've got pre-existing immunity, there's always a preference for that pre-existing immunity in a way, right? Because you've got existing B cells that can rapidly expand. So we see this all the time in flu, right? People will mount a big response to what they've seen before because that's kind of the army that's ready to take it on right away, while you're then generating a response, hopefully to that new exposure. And so one of the things we thought was like, well, maybe we're looking too soon. Maybe 30 days isn't long enough. We should look at 90 days. So we looked at 90 days, and they still looked very similar at 90 days. Maybe that you need a second exposure. It may be that there's so much immunity that was already generated from that original vaccination against the original strain, that if you give a bivalent booster, something that has that original virus that original antigen in it. It doesn't have the virus. It has the antigen, right? It may be just so hard to overcome that bias that your immune system has towards the original antigen. But that's something that we and others are working on to try to figure out to help hopefully make better vaccines, or at least provide some advice on how to do that. So I'm going to shift gears. Am I doing okay on time? Yeah. Okay, yeah, I should be able to finish in 10 minutes. And talk about something different that we're looking at in the IASO study, so post-acute sequelae of COVID-19, aka long COVID. This is data up here from a recent CDC survey called Pulse that really was to characterize the burden of long COVID or PASC. So in that survey, they found that 31 million US adults had ever had symptoms of long COVID, and 12 million US adults currently had symptoms of long COVID, so really a huge burden. It's a disease or syndrome that's hard in a way to study because there are variable symptoms and really variable severity. But some of the things that are common are people might have lingering respiratory symptoms, they may have fatigue, report fevers, report cognitive issues. So to look at long COVID or PASC, we used IASO infections that occurred and throughout the entire study and those that occurred prior to enrolling in the study. So we looked over this entire period, and what we found was that PASC was less common in vaccinated individuals. So we used two different definitions, PASC at 30 days, so that's the CDC definition that they're using. The WHO uses 90 days, and we decided to go with 90 days because we felt like it was more clinically significant. Certainly I have had infections in the past, a respiratory infection where 30 days out, you're like, I'm still kind of coughing, right? And so we decided to go with 90 as a primary outcome, but also look at 30. So when we looked at 90 days over the entire course of the study, 8% of individuals that had been vaccinated at the time of their infection reported that they had at least one symptom of PASC or long COVID. And that was compared to 27% of individuals that were not vaccinated at the time of their infection. One symptom is, of course, not great. Having a bunch of symptoms is perhaps even worse, right? So we also looked at having 5 or more symptoms, because some people are reporting a lot of symptoms. So for 5 or more symptoms, overall about 1% of people who were vaccinated at the time of infection reported 5 or more symptoms 90 days out, compared to 4% of individuals that were not vaccinated at infection. And both of these were statistically significant. So we have here one, this over here is on the left of one, indicating that we saw a lower prevalence of PASC in vaccinated people, and that vaccination was protecting against long COVID. Of course, we don't just care about if you have any symptoms, we also care about the severity of those symptoms. So we categorized that, looking at max severity. We also looked at average severity. And there, once again, at both 30 days and 90 days, we saw that individuals that were vaccinated at the time of infection, if they did have symptoms, those symptoms tended to be much less severe than people who were not vaccinated at the time of infection. So one of the problems with this, that's great, good news, right? But one of the issues with this analysis is that most of the vaccinated infections are happening, a great proportion of them are in the Omicron era, and we know that Omicron is less severe, at least in the acute phase. And so we wanted to go and look at this phase before the introduction of Omicron. We have less power to do that, but we thought it was important because the variants do differ in severity. So when we did that, we also saw that PASC was less common in vaccinated people pre-Omicron. So here you can kind of see it was 18% in vaccinated people pre-Omicron at 90 days, compared to 28% in unvaccinated people. I will note that this is not quite statistically significant, in part because of those lower numbers. The vaccine prior to Omicron was still quite good at protecting vaccinated people from getting infected. So we had fewer infections in vaccinated people during that time period. We also looked at severity, and there again, we did see that PASC was less severe in individuals that were vaccinated at the time of their infection, and this is all in the pre-Omicron period compared to people who weren't. So we did one last comparison because we were wondering, like, well, based on those two, it looks like it's in Omicron it's probably less severe, so it'd be nice to get a handle on those numbers and see what they look like. And so we looked only at breakthroughs this time, so only vaccinated individuals, and we're comparing people during the Omicron period to the pre-Omicron period. And so during the Omicron period, about 6% of the cases that were detected, or the infections that were detected, reported symptoms at 90 days, compared to 18% pre-Omicron. And so that was statistically significant, at least for any symptom. For both groups, they reported a very low prevalence of 5 or more symptoms, so 1%, so there was no difference there. When we looked at severity, however, we did not see a reduction in severity for the Omicron compared to the pre-Omicron. There may even be a little bit of an increase, hard to say with the numbers because they were fairly low, particularly for the 90 days. But we saw a reduction in the number of people with long COVID, but those with long COVID were not less sick than in the pre-Omicron era. So in conclusion, IASO was established pretty quickly to address critical questions about SARS-CoV-2. The team did an amazing job of getting this up and running. I think we were enrolling participants within a month of receiving funding, believe it or not, and really rapidly ramped up to get to almost 3,500 participants. It's a partnership between Michigan Medicine and the School of Public Health, and I think a very successful partnership. It plays a critical role in monitoring of SARS-CoV-2 variants as they arise. And in that analysis of long COVID, we did find that PASC was less common and less severe in vaccinated individuals compared to unvaccinated individuals. So even though the vaccine isn't necessarily as efficacious at preventing infections at this point, the good news is that you will have reduced risk of long COVID symptoms. So we have a lot of ongoing work. We're looking at vaccine effectiveness, and we are really uniquely positioned to deal with some of those biases that may be occurring in the estimates as well. We're looking at the response to reinfection, PASC and reinfections, the effect of first exposure on future immune response, because there's some concern that you might see imprinting where your first exposures then forever change how you respond to SARS-CoV-2, and that can have limitations potentially for how protective the response is. And so there may be some, you'd want to design vaccines to get around that ideally. We're also looking at social distancing behaviors and how they changed and how people kind of responded to messaging that went out to the public to inform future responses. And then we are continuing to monitor variants as they arise, and I should say that we will monitor new vaccines as they roll out as well, or as the vaccine gets updated. And with that, I'd like to really thank the study team, in particular Rick Valdez, who was kind enough to lead this study with me and has been fantastic to work with, Carmen Garciam, David Menthai from Pathology, who are co-investigators on this, as well as Emily Stoneman, who you may know heads up OHS and participates in this study. And then our amazing investigative team, everybody on the SPH side, as well as on the pathology side. This really is a huge amount of work and has taken an incredible amount of dedication to set this study up and run this study throughout the pandemic. The funders, NIH, and especially the participants, and maybe some of you are participants, I'd like to thank everybody who participates in the study. And with that, I could take questions if there are any. So at the beginning of the pandemic, there was a difference between races.
Yeah. So my question is, do you see any difference about the vaccinated population, depends on the race or age, than they say different, like the production of the antibodies or something, or is, because I noticed that you present data is like a vaccinated group.
Oh, yeah, so we do present, we have looked at that, and we don't really see much of a difference. The vaccine has a lot of antigen, so it really generates a robust response, even in individuals that are quite elderly. I would say we don't have any like frail elderly in our study, so that would be a different group who would probably be at more risk. I think, most of the differences, maybe not all, but most of the differences, racial differences, are probably due to social and societal factors. So they were at greater risk of being exposed. They may have been at greater risk of being exposed in higher dose, which was probably important. And they, in general, may have less access to healthcare or good healthcare. And I think all of those things, unfortunately, came in to play early on in the pandemic to create some huge inequities.
Okay, thank you. So, in the selection of your participants, Dr. Gordon, did you also consider participants having underlying disease, or they're altogether, like, I don't know, how selective were you with your participants?
So we only excluded people if potentially like participating in the study would be a risk to them. So we have people who are undergoing chemotherapy, people with immunosuppressive conditions. We have people with allergies who can't get the vaccines in the study. Diabetes, obesity, just really kind of generally representative. One great thing about having a study of this size is we have kind of more ability to do so, right? So if you have a really small study, you start to think like, oh, well, I've got to make the population as similar to each other as reasonably possible, so that we can have more power to address these questions, because we don't want to exclude people who are immunocompromised, right? Not be able to count them in this main analysis. We didn't need to do that. So really it was, I think predominantly bleeding disorders, and I think, we did have discussions with potential participants if they brought up something, and we were like, sounds like you're already going in a lot. Are you sure you want to go in for additional blood draws and commit to this? We didn't want it to be too much on the participants, but that was it.
I'd really like to thank her again, and if we could give her another round of applause for coming here to speak. We'd also like to present her with this paperweight bookshelf memento, a small token of our appreciation for you and your partnership. It's been very great to work with you, and we look forward to trying to complete all the things on your list there. Thank you.
Okay, great, thank you so much, Dr. Gordon. I have the session code for your PACE credit, and it is 7821, 7821. So please grab a lunch if you haven't had lunch yet. There should be some drinks and snacks brought out. Enjoy the exhibits. And our next talk will be at 1 o'clock. Dr. Joseph will be talking about molecular diagnostics.